today 5 o'clock, 5 p.m. And we start our laser meeting, Laser Talks Brussels, devoted to the light, color, and matter. And today we have 3 speakers with us. This is Adrien Lucca, Sebastien, Mouchet, Sintobin, sorry. Edith Doove, she is as usually with us, and she will moderate this talk. And we will try to discuss as much as possible this interesting topic about color, light, perception, and how we are more or less influenced by this, and how we can do influence in other things and topics and matter. So Edith, I would like to give you a floor. So.
Okay, thank you. And welcome to the speakers and our audience. So the topic light, color and matter is an exploration into the dynamic relationship between perception and materiality. It encompasses a broad spectrum of topics, including the study of vision in various organisms, from humans to animals and insects, the fundamental properties of electromagnetic radiation, the behavior of light waves, and the interaction of light with matter. Exploring texture, transparency, reflectivity, and other material qualities enriches our understanding of how light interacts with surfaces and shapes our perception of the world. Because this topic is vast and could touch on many subjects, we will limit our discussion tonight, this late afternoon, mainly to the perception of color and how we learn from or can be inspired by the animal and plant world in this respect, which touches on biomimicry. But let me first introduce our speakers. So to start with Adrien Lucca, and that will also be the order in which they will present. Adrien Lucca, since 2009, develops a multidisciplinary body of work around color and light that questions our perception of the physical world. In search of practical means of action to set up aesthetic experiences, he has set up a research and production laboratory where he conceives his work in an autonomous way at the intersection of art and science. To the antipodes of a sad passion for the normalization and the technicization of our relations to the physical world, Lucca believes that one can highlight the strangeness of the link between the physical world and our perception of it by appropriating scientific and technological resources. His most recent work aims at redefining the very concept of color. He lives and works in Brussels, was born in Paris, and is currently in Paderborn in Germany, where he works on a new project. So his studio, created in 2015, is a Belgian company whose main activity is the design, production, and installation of monumental artworks in the public space. The studio has in-house research, innovation, prototyping, and production activities that allow it to be largely autonomous with regard to the projects it undertakes. The main specialities are pigment color, glass, and LED light, which are used in an approach that combines chemistry, physics, and perception. Projects are therefore either entirely made in-house or developed via a fleet of digital fabrication machines before being outsourced for production. The studio is equipped with CNC, 2D, 3D cutting and printing, physics and chemistry devices, custom designed software, and a paint formulation lab. The studio has collaborated with many specialized companies, glassmakers, glass factories, paint manufacturers, industrial production of metal structures, specialized lighting companies, custom electronic component manufacturers, etc. The studio has thus acquired considerable experience in the realization of large-scale projects. Dr. Sebastien R. Mouchet received his BSc, MSc, and PhD degrees in physics from the University of Namur in Belgium. Since his MSc and PhD degrees, he has studied the physical origins of various visual effects evolved by natural organisms, from additive colors in photonic polycrystals and fluid-induced color changes to fluorescence emission in natural photonic structures and UV and IR scattering in integuments. You have to explain what that is. later. After the completion of his PhD degree in 2015, he worked as a postdoctoral researcher at the University of Exeter in the UK for 4.5 years. He's now carrying out his research with the University of Namur as honorary lecturer and researcher, senior honorary lecturer and researcher. And with Professor Olivier de Paris, he co-authored a book entitled Natural Photonics and Bioinspiration in November 2023. He was awarded the 10th Charles Courtois Prize for the best PhD thesis in physics of the University of Namur for 2014 until 2017. And Heleen Sintobin has a master's in design products at the Royal College of Art in London. In her current personal work, she's exploring the future of crafts in the digital age. Heleen also works as an artistic researcher in Kask's Biolab Laboratorium in Ghent in the interdisciplinary project Ecology of Color. And together with scientist Mario Botus, she explores the future of sustainable color production. And although her bio is considerably shorter than the others, that doesn't mean she's not interesting. Otherwise, we wouldn't have invited her. So I suggest, Adrien, that you start your presentation.
Yeah.
So of our speakers will present for about 15 minutes approximately, and then we'll then I've concocted some questions for them, which they will try to answer. Up to you, Adrien.
So I share the screen like this. So hi. So I will mostly discuss one research that I'm developing at the moment, because it's a new topic. I think it was the most interesting thing I could be talking about today. And so I'm going to start by very quickly say what I do. And then I'm going to directly talk about this research, which I called Parallel Worlds, which is about like the vision of human versus the vision of a specific insect, and how can this somehow make us think in a different way about light and color. So I'm an artist. I do, as it was written in my bio, I do mostly like monumental artwork in the public space with glass, but I'm also a lot into geometry, mathematics, computer programming. I also collect pigments and colored materials, mostly synthetic. I work also with LEDs. I create light sources. I work also with glass in a more traditional way. I do some stained glass works as well. This is a sample of something that is currently being made just behind me right now. I will talk quickly about it at the end. So my practice is entirely revolving around light and color, and so I just want to remind you that light is defined for human beings as the part of the electromagnetic spectrum that our visual organs are sensitive to. So we are, as human beings, most of us, we are sensitive between 400 and 800 nanometer. And inside of this range of energy, there are these colors that everybody know that appear, that look like very similar to the colors of a rainbow. And when I use pigments, I combine the spectrum of light and the properties of the colored material, which are most of the time differences in terms of absorption and reflection of this light that is coming on the material. And there is one concept that is important in color science when it comes to the effect of light on the color of the materials. And this concept is called color rendering here. So on the online, you can find many pictures like this. Most of them are completely wrong, but So you would see, for example, if you buy a lamp, you have a CRI, which means color rendering index, that can be 98, so that's like very good, or 70, this is like very bad. And you see like scales like this, which most of the time do not make sense, but this doesn't matter. The concept is that depending on the quality of your light, your colors will appear differently. Your red may be more red or less red or too red, etc. And I'm working on this aspect of light. So, for example, here, this is like from a small paper that was published in a conference a few years ago. On the left, you have like a curve that represents natural light and a few color samples, and you have a picture with some real objects. And on the right, you have like another type of light spectrum, which here is a kind of theoretical thing. And you have a picture that shows how the objects would appear if this light was used in this scene. And you can see that in this case, all the yellow objects become red, and the blue disappears. Is like the ultramarine blue becomes like a kind of turquoise blue. So I've been working on this. I created the devices. I call them light synthesizer or programmable light synthesizer. They're based on LED lights, and I can tune the spectrum of light in order to make the objects change in color. So for example, in this installation here, the same balloons are bright orange on one side of the space, and on the other side, they become yellow. The interesting thing is that when you work only on this aspect, on color rendering, you can do it without changing the color of the light itself. So the effect only appears on the objects, but the light doesn't seem to be different on one side or on the other side of the room. And this is, of course, very important in order to achieve this kind of magical experience as in this installation. So I've been working on this, and I was searching for many different types of materials. I was searching for specific properties, and there were some properties that I couldn't find. So I decided to check the colors of flower petals. So this is, for example, a page, an image of the spectrum of reflectance of a certain type of flower that can be found on this website, reflectance.co.uk. This is a database where like biologists have measured the reflectance or the color, if you want, of thousands of flowers. And I thought maybe because the colors of the flowers are not selected by human beings, but they are selected by insects over millions of years of co-evolution. Then I thought because the insects have a different visual system, maybe the properties of the dyes and pigments in the flowers, maybe they're a little bit different. So I started to make some experiments, changing the colors of flowers with light. And so it became a sort of installation in 2022. And so recently I got a scholarship to develop this further, and I started to read more biology and trying to understand like the visual system of some different species, insects, etc. So for example, there's like a very nice article here where we can see that in the tree of evolution, there are many different kind of visual systems. Some individuals have 3 photoreceptors or less. Some have 4, 5. Some butterflies have up to 16 different photoreceptors that are sensitive to, or that are like used to code or to sense color. So when we get out of the human definition of color, we end up completely somewhere else where there's such a diversity of visual systems that we cannot anymore define color only relatively to the way that humans experience it. And my interest, what I wanted to question mostly was like, so the entire light technology that we use and put in cities and around us is calibrated or designed accordingly to the human visual system. And so I was wondering, okay, how do the other species somehow perceive our colors and our lights because they are made for us and not for them. So I found this article in Nature Communication, which is interesting because it's really about this. So this article studies the color vision of this insect, which is called an elephant hawkmoth. And this insect has this specific characteristics that it's very good at seeing color even at night. So even just under the light of the stars, this insect will still see colors around in the environment. And the aim of the article was to study the effect of different types of artificial lights on the color vision of this insect in terms of camouflage, in terms of communication between individuals, and in terms of does the insect see the flowers that the insect is pollinating. And so this article had some results for different, comparing the moonlight with different types of artificial lights. And in the table here, you have the results, which were saying basically that low pressure sodium and orange LED, which are like some type of lights that I will present, are very bad, and the others are like kind of ranked according to the possibility for this insect to see colors. So one very important element that differentiates the different species is the sensitivity of their eyes. So here on the left, you have the moth. It has like 3 types of cones. So this, as we human beings also have 3 types of cones, but the sensitivity of the cones is different. There's one cone that is sensitive in the UV, where we are blind, basically. Then there's a short wave, blue, and medium wave, which is green, while we have blue, green, and red. So we see, for us, we see red. For the insect, red is black. It doesn't see black. For us, UV is black. For the insect, UV is something, is UV. And so in the article, like these natural lights, which have a specific spectral distribution of energy. So for example, this is the moon, starlight with many spikes from different types of stars. So these artificial lights are compared with, natural lights are compared with artificial lights. So mercury vapor, which is a traditional street light, metal halide, which is another type, low pressure sodium that we have on the Belgian highway, PC amber, which is a type of orange broadband LED, white LED that we see now everywhere, etc. And for me, there was something really odd in the results of this article, because the article says that white LED is very good at rendering colors for the moth. But if you check the spectrum on the left here of white LED, which is the black line, against the sensitivity of the cones of the insect, you see that because there's no energy in the UV, it doesn't make sense that the color rendering of this light source is good, because it's the same as if we were lighting our office with a bright yellow light made of red and green. We wouldn't see any blue in the environment. So because that was so weird, I started to you see like the cone response to this light is 0.8% of the total, so it's very little. And so because of this, I started to re-implement the entire code of the article and exchange emails with the authors and check all the data, reproducing everything with different methodologies. And I think that now it's pretty clear that there is a problem with this model that they have been using, but we still don't really know what is the problem. But I think we're gonna figure it out. I want to show you some pictures of an exhibition which was in a museum in Belgium last summer, because I think that what you can see sometimes in my artistic work would be pretty much what the insects are experiencing in terms of distortion of the color rendering of for example, their ability to see a flower against a green background and things like this. So what I do is, for example, I have some pieces of, some pieces of glass like this on the floor on a gray background, and with the light they change in color like quite strongly and smoothly. And if you're an insect and you are coming from like a dark area, which is only lit by moonlight, and you arrive in an area where there is a human artificial light, then you'd probably, in terms of color, you'd probably experience something a bit like this. Sorry, I'm showing it again. A bit like these glass blocks here that become black or yellow, and then they turn completely, sorry. And then they turn completely red, for example. And then you don't see any kind of color difference between different objects. And in my opinion, this should be, we have to verify this, but this should be much more like what our light made for human beings does to the visual system of this hawk moth. So I'm starting to think this project in terms of almost like a video game. So I want to make a spectral study with photography of the insects themselves or of their environment. And I want to understand this better and make a kind of installation where I would play with light and try to make us experience this kind of distortion of reality that, in my opinion, they are experiencing when they are getting closer to our light. That guy is the guy I'm discussing with because he's one of the authors of the article, and he has also like another. He's developing a digital toolbox to process photographies with the like the resolution, the angular resolution of the eyes of different animals. So there's also like this possibility of creating shapes that are, for example, here in the bottom right, you have like a small bird, the same flower to a small bird, a house sparrow looks like a kind of very simplified shape. And I want to dive into this as well. Yeah, so because I have only 15 minutes, I think it's better that I let the other speakers speak and that we exchange more in the Q&A part.
Yeah, okay. Thank you very much, Adrien. It's very interesting, and I have indeed some extra questions. So, Sebastien, if you can give your presentation, please.
Of course. I'm trying to share my screen. Well, that's a pleasure for me to present some of my research and also introduce our viewers to light, color, and matter, which are actually three terms that corresponds also to my research at the University of Namur as well as the University of Exeter, as Edith mentioned earlier. So, well, the first concept, of course, to clarify, Adrien obviously mentioned it as soon as my PowerPoint will allow me to move to the next slide, which, is indeed the concept of light. What is light? And Adrien indeed mentioned that it's an electromagnetic wave. That's a model that we use as physicists, but we also describe light as a set of particles that are called photons. So that's indeed a wave, as I mentioned, like the wave that people can observe at the sea surface, for instance. But instead of having a mass of water oscillating, these are two different physical quantities that oscillate, an electric and a magnetic field. And I will use much more that concept in this presentation. Adrien mentioned also some wavelengths, the wavelengths that's the distance between two maxima of the wave. And as Adrien also mentioned it, as human beings, we see visible light, which is only a tiny part of the electromagnetic spectrum. But I'm sure that our viewers know many other types of electromagnetic waves with different wavelengths, such as infrared and UV light that Adrien mentioned, but also microwaves or X-rays. The thing is that color, as we already understood from the previous talk, is not only a wavelength. It's much more than that. That depends on the sensation that we have, but also the perception that we have. You probably know, if you remember your classes in secondary school, that on the retina of human eye, there are some cells, rods and cones that are sensitive to visible light, and the cones are the ones that are known to give rise to chromatic vision, the vision of colors. Problem is that we don't perceive colors the same way. So, for instance, Edith is wearing today a pink pullover, if I'm not mistaken.
Yes.
Let's assume that the meeting was in person, so there would be no screen in between us. That's very unlikely that there would be any two of our viewers would be able to perceive the same pink. There is some subjectivity in the color perception. Another example is that color. If I ask our viewers what is that color, probably some people will answer green, other people answer blue. Probably at some point we may agree that this is called turquoise, but well, that's another example of subjectivity of colors. Well, colors requires 3 or 4 different concepts. First of all, light sources. Adrien mentioned the issue with light sources. Well, a light source could shed some light on an object. That object will reflect back, scatter back some light in the eye of a viewer. That viewer will be able to detect that light thanks to the cells that I've mentioned. By the way, we can also perceive color directly looking at the light source. We don't necessarily need an object. And when we've detected the light source in our eye, well, our brain processes the information and give rise to the color perception that we have. So Adrien mentioned some very beautiful examples. I'm probably sure he doesn't find the high quality or the quality of my pictures specifically high, but just to explain the point where we have here a picture of a green leaf and a red pepper, and they are illuminated with different colors, basically color light. With some green light, we can see the pepper red, and the green leaf with the red light is black. With blue light, both objects are black, and with some green light, only the leaves appears chromatic, green in that case, and the pepper is black. That's exactly the phenomena that were described by Adrien. I also prepare another example from an app on the internet where we have a flag that is illuminated by some red light. We've got, well, some colors that by coincidence corresponds to what more or less to the flag of the province of Namur, if you're familiar with that one. If to the red color we add some green light, well, we get some Belgian flag. And if we add some blue light, we get a French flag. Of course, that website is French, you understand that. But, well, of course, the color that we perceive do depend on the illuminant. I hope that you understood it now, if you hadn't understood it from Adrien's talk. And so these problems, in a way, the subjectivity of color perception, that I don't perceive the same colors as Judith or as Adrien, that colors depends on the illuminant, are somewhat problematic, I would say, in science, in hard science, in physics, because we like to quantify things and we can't have quantities that are subjective. And so fairly early, there were some color spaces that were developed. One of the early one was by James Clerk Maxwell. So it's Maxwell's triangle, and it defined, well, a triangle where one color was represented by a set of coordinates, RGBs. And actually, these coordinates are the ones we still know for computer screens. I mean, they are defined somewhat differently for the computer screen, but the origin dates back from that. Nowadays, we, of course, use other chromaticity diagrams. Adrien briefly mentioned some or show some. One that is fairly used in my research field is the CIE chromaticity diagram from 1931, where each color is represented by a set of coordinates, coordinates X and Y, for different illuminants. Adrien showed you some curves corresponding to different model for the sun, basically, or light sources in general. So what are the origins of colors? Well, there are three. We mentioned clearly light sources, but there are also pigmentary colors and structural colors. So pigmentary colors, they are due to some pigments. These pigments absorb partially some light that would be shed on them, typically white light. That energy is transferred to the material as a heat, or it could be also as a chemical energy, for instance, for photosynthesis. And light that is not absorbed is scattered back in the environment. So some examples are organic pigments, but there are also artificial pigments, usually food coloring, or mineral pigments. And pigmentary colors are almost everywhere in a living organism, in mammals, in birds, in insects, and as well as plants. The second type is, well, that I decided to present you is light sources. Well, you're surely familiar with LEDs or LEDs, light screens, of course, light bulbs, fires, but also bioluminescence. In the case of fireflies here, of course, stars, we mentioned the sun earlier. One type of light emission that is interesting for my research is fluorescence. Fluorescence is the emission of light by a molecule called fluorophore, following the absorption of incident light that is more energetic. Typically, some UV light will be absorbed by a pigment that will re-emit it in the form of visible light. UV is more energetic than visible light. And that phenomenon takes place in fluorescent tube, for instance. Tonic water is known for being fluorescent, and you're also probably familiar with anti-counterfeiting technique for banknotes or passports using UV light. So we have living organisms are often fluorescent. That could be the case of insects. So here I show pictures of insects under visible light on the left and under UV light on the right, and they emit by fluorescence. Also, sea animals are fluorescent, such as here jellyfish, but sea anemones. Actually, we don't need to go very far in the tree of life to find some fluorescent organism. Us as human beings, we are also fluorescent, or nails are fluorescent, or teeth are fluorescent, as well as white hair are fluorescent in human beings. So structural colors. Structural colors involve light interference. So to have some light interference, we need at least two waves. Let's assume these two waves here, where the maxima of one corresponds to the maxima of the other one. they interact. And this interaction gives rise to a wave that is simply more intense because they will combine with each other. And such interference is called constructive interference. However, if we assume now that one wave, that the maxima of one wave corresponds to the minima of the other wave, when these two waves are summed up, well, basically they destroy one another. They cancel each other, so we get nothing, and this is called destructive interference. And of course, light can interfere according to many other cases that ranges between these two extreme cases. So let's assume now some light interacting with a film of oil, for instance, that we can observe on the floor. So that wave will first interact with the first interface of the film. Some light will reflect. Some light will transmit into the film. That's called refraction. That light will then interact with the second interface here. I hope you see my mouse, and will reflect on the second interface. It will also refract through it. I even represented this in this figure. The light that is reflected will then interact again with the first interface. Some light will be transmitted, some will be reflected, and so on and so on. And so we've got here waves that are somewhat delayed with each other, and that give rise to the light interference I mentioned earlier. Typically, the dimension of the film should be in the order of 100 nanometers. So if you are not familiar with nanometers, let's say that if you divide the diameter of a football by the diameter of Jupiter, you get the same ratio as if you divide 100 nanometers by the diameter of a football. So long story short, it's fairly small. And structural colors are present in our daily life, maybe not every single day, but regularly. So bubbles are examples, CDs, DVDs, if you still have some at home. Stained glass windows, I mean, some stained glass windows will be colors by structures, rainbows, etc. They're also present in natural organisms, such as some fish, some birds, some mammals too, insects, of course, and also in some plants. One property of these photonic structures is that they are usually iridescent. That means that the color changes with the angle of observation or the angle of incidence. Here, there are 6 pictures of the very same beetle observed with different angles. So the 3 top pictures are dorsal views and the 3 bottom pictures are ventral views. And you can see that depending on the angle, we observe various colors. Another example is that GIF. We have here morpho butterflies that is rotated around a rod, and you can see that the color that is observed changes from violet to blue depending on the angle. So the structure that gives rise to that color, or the color of that beetle, that is called a jewel beetle, by the way, is a multi-layer structure of different layers with different density and different optical properties that give rise to the light interference I explained earlier. In the case of the morpho butterfly, the wings are actually covered by some scales that we can see here. And actually, if we look at cross sections of these scales here, the B figure, we can see some structures that as physicists we describe as Christmas tree structures, because for physicists, this looks like a Christmas tree. Anyway, these structures also give rise to light interference. The process is slightly more complex than the thin film I shown earlier, but it gives rise to the blue colors that we observe with this butterfly. A last example from my research, somewhat more recent, is the backscattering of ultraviolet by eggshells. So with a student of mine, we analyzed the optical properties of eggs from hens, but also from ducks and kills. And basically, we found that there are some peak of reflectance, of scattering, backscattering in the UV part of the electromagnetic spectrum. So fairly close to the UV, but not quite in the UV. And we try to explain that. We look with electron microscopes at cross sections of these eggshells, and we observe the presence of pores in the structures. There is a layer here that is about 200, 220 microns thick, so about 0.2 millimeters, if you are not familiar with microns. And within these porous regions, we can observe all these spherical pores, so basically bubble of air in the material of the eggshell. And these pores, we demonstrated with measurements and also simulations that they give rise to an extra UV backscattering reflection, if you want, in the environment. To give a metaphor to explain the phenomenon, it's very similar to what happened with clouds. If you look at dark clouds, as we are used to in Belgium, these black clouds that we see from the ground, not exactly today, but you know what I mean, right? Well, they appear black because light that comes from the sun is actually backscattered by these tiny droplets of water that are in suspension up in the air. If you are lucky enough to be above these clouds, actually these clouds appear fairly white. I mean, they are bright white actually, and that's due to such backscattering. And the phenomenon is very similar for the UV backscattering in the avian eggshells. The dimensions are slightly different that explain that it's active in the UV and not in the visible. And one possible use that nature found of this backscattering is actually the protection of the fetuses in the eggs from UV light that could damage the DNA of the fetuses. So, well, explaining this phenomena is nice from an optical point of view. That could also give some insight on some biological functionalities. But we can also use the knowledge that we acquire observing nature to develop new material, combining it to the knowledge that we have in terms of material processes, let's say, to create new novel applications. One application that I recently developed in my lab was actually bird-safe windows. Well, you may know that birds every now and then strike windows, and actually such collisions are fairly detrimental for the life of birds. In most cases, the bird will die. If not within a few minutes, it may take several hours due to internal lesions, actually. That's actually the second cause of death of birds on Earth, right after cats. And that's not only crucial for some species that may be already in danger. As owners, as insurance companies, that's not always great when we have to replace a window on a car or on a house, for instance. Well, there are some solutions that exist. You probably know the stickers there of birds. According to birds conservationists, they are useless, to be fair. I think that my opinion that there are some anthropocentric representation of birds, and birds may not perceive this as a threat to them. But there are other solutions that are effective. Well, to place some nets, some curtains, some stickers, some screens, some grids, whatever pattern you want, inside, outside, within the windows, that works. Problem is that, well, usually we like our windows to be transparent, right? And so I took inspiration from dead butterflies that actually we find in Belgium, the purple emperor. It has some violet reflection here that you can see on its wing there. That is due to some structure occurring in its scales here. You can see the Christmas tree structures that I mentioned earlier for the more for butterfly. The dimensions are slightly different, so that the color is more in the violet. And actually, these wings, they don't appear violet because the reflections takes place in the ultraviolet. Ultraviolet, that is perceived by birds that have an extra cone, as mentioned by Adrien earlier. They have four cones, and they are able to perceive ultraviolet, unlike us human beings. And so we can take inspiration of such a reflector to make windows that is perceived by birds. Also, we can create some color space for animals. We know the optical response of the cones. For instance, in the case of dead fish, that's a study that I've made with some colleagues in Australia and student in Namur, and we can develop similar color space as we did almost 100 years ago or more for human beings. And that's what we did in the case of that species to represent some colors perceived by some fish species. So, well, back to that bird-safe windows. I decided to take inspiration from that butterfly's wing, simply approximating the structure by a periodic multi-layer that is transparent for human beings in the visible range, but that is chromatic for birds, while taking it into account accurate vision models for both human beings and birds. So, well, within our physics department, we deposited such coatings that were developed with of course some computer simulations. And here are some pictures of some samples that were produced, where you can see here on the left some devices under visible light with a visible camera with some white and black background, whereas there on the right side you can see exactly the same devices, the corresponding devices, under UV light with a UV camera. And there we can see a pattern that is visible by birds. So that was a recent application that we developed. So in conclusion of this talk, I would like you to take away, but you are possibly already home, so it's not a take-home message, but more a stay-home message. That light is both described as a wave and a set of photons in physics. That colors originate from pigments, but also light sources and physical structures. And these structures are very sophisticated materials that displayed some interesting optical effects. And studying them in nature not only help us to elucidate some physical phenomena and also the related biological functions, but also help us to develop novel applications through a so-called bio-inspiration approach. So thank you for listening, and don't hesitate to ask questions about this research during the Q&A sessions.
Thank you, Sebastien. Again, really intriguing and very rich. So now I would like to invite Heleen, if you can stop sharing your screen.
Hi everyone.
Yeah.
I would like to tell you something more about the artistic research I'm doing together with my colleague Maria Boto and Amandine David at Laboratorium in Castres University College and University College in Ghent. Ecology of Color is part of Laboratorium, and Laboratorium is the art and design biolab at Castres. And the nice thing about Castres is that there is space for artistic research. So back in 2016, my colleague Maria was asked to kind of start a biolab, and she quite soon decided to focus the research on color, because color is kind of an interdisciplinary language between all the different courses in the school. So everybody can come to us. Students are always welcome in the lab. And our specialty is sustainable color production. And this picture kind of says it all. It's a picture of the Ganges River, where you see the factories in the background, but also a colored, polluted river. So there is a lot of things that go wrong due to color pollution because of the textile industry and fashion industry. In the research, we have two research lines. The first one is called Living Colors. That started in 2016 into research of traditional pigments and minerals, mainly from plants. But then quite soon, because this method is quite traditional, but also quite expensive. I mean, you need to grow the plants, you need to harvest the petals of the flowers and so on. So it's quite expensive. Maria decided to make the jump towards biotechnology, more specifically into growing algae in the lab and also growing bacteria. Both of them are connected to pigments and colors that you can harvest. And the advantage as well of especially microalgae is that you can upscale. So first of all, you can control it in the lab, so it's not dependent on weather like it is with plants. And then there are also algae farms that can upscale the process and where you can obtain much more pigment. So these are some images of the database of the natural pigments. We also have a physical database in the lab. But then, yeah, here we are with the algae. So what we do with the algae is we grow it, we feed it. You need to take care of it. It takes a couple of months before you have enough biomass. Then you sieve the biomass, you collect it, and then you put it in the freezer where you break the cells so they start releasing pigments. And you would think that the color palette of algae is quite brownish, bluish, greenish, but actually there are also pink algae, and these are all the pigments that you can obtain from different species of algae. And then, of course, when you start to mix them, you can get whatever color you want. So students are not disappointed when they come to our lab and they want some colors. So we are based in an art academy, which also means we need to look into applications. So we developed a recipe for screen printing, different colors on top of each other, for example, with different natural binders. We also did a small collaboration with a book company to develop the covers of those notebooks with screen printing. And then also, yeah, the more, the pen plotting. And then during the process, like once you apply the pigments onto paper or textile, we discovered that the colors starts fading at a different rate. So depending on the type of algae, the type of color, the colors start fading at different rates. So for example, the yellow here already starts to fade after a couple of hours in the sun, while the pink stays for a couple of months. But we, yeah, in the beginning it was kind of a disappointment, but then we tried to look at it as a kind of smart ink, quite primitive one. But could you, for example, develop a time-based color palette? And then, for example, in graphic design, look into posters, applying different colors, and then the patterns would start to change over the time. So we tried to approach it that way. These are some fading tests in industry lab. Some screen printing. For an exhibition, we made a really big calendar of 30 meters long, 6 months, and every day we hang one part of the calendar, and then you could see kind of a live, a real-time fading effect throughout the exhibition. We also applied the pigments to PLA, the 3D printing substrate. And there we saw that some algae kind of fired because you need to heat it to 200 degrees, and they left some ashes behind, which is also kind of a nice effect. but the orange worked quite well, but also is fading over time. So this is one research line, and then the other research line is what Sebastien already mentioned in his presentation, is about structural colors. For that, we collaborate with Northwestern University and the University of Akron from the US and the University of Ghent. So if you look at the visible light spectrum, the upper birds, for example, they have their color because of pigments. And that means that depending on what they eat, they show their color, and they also can show to the female birds how healthy they are. But then the lower part of the birds, they have pigments and structural colors on top, and you can see that's a way broader fields in the visible light spectrum. And that's also why it's for us very exciting to move from pigments to structural colors, because it's a way bigger field. So the structural colors are these kind of metallic, shiny colors. You can find them in beetles, but also in birds. In nature, they're mostly used for camouflage, but also for mating. So mostly it are the male birds that look the most beautiful to seduce the female birds. So yeah, often it's a mixture of pigments and structures, but this is, for example, an image from the electron microscope, and there you see the 3D structure. Sebastien already mentioned the iridescence. So basically, depending on how the light hits the 3D structure, the nanostructure, then you see a different color. So depending on the angle. This is some zooming to nanometer scale, 500 nanometer scale. This is a wonderful color world of butterflies. And here you see as well the Christmas trees Sebastien mentioned. You also can find social colors in plants. For example, this is the African polioberry. And then it's not melanin based, but it's cellulose based. So the structure looks slightly different. And what we do in the lab is we grow structural colors from synthetic melanin. So basically what we do is we put together two chemical components that react and that create melanin. And melanin has the capacity of self-assembly. So basically what will happen is they will react chemically, then they will create little balls. The little balls will flow up to the surface. And then depending on the recipe we use, you get a different thickness of a film. This film starts to appear already after a couple of hours, but is fully grown after 1 week. And then you have a range of different colors. In total, we have 12 different recipes at the moment. So a scala of 12 colors. And then it's like, trying to scoop this film with some material. So this is, for example, a little glass plate. This is just a film on glass. And here you see, this is the melanin. So melanin is a brown pigment. But then depending on the angle that you hold it, and depending on how the light catches it, you see the colors. This is like the iridescence I talked about. So the blue becomes purple or dark blue. The purple becomes orange. Here's some more. And the nice thing about it is that there are still happy accidents happening. So we know more or less what the recipes will end up like, but we still have crazy colors coming out as well. Also, when you start to play around with scale, you also get different results. This is an analysis from the spectrometer. And this is an electronic microscopic image where you see the film and the little balls of melanin that define the thickness of the film. Then again, looking for applications. So we tried out different materials. So this is some textile, polyester, PLA, ceramics, different types of paper, photographic paper, glass. And every time the material we apply the color onto also sometimes chemically reacts with the melanin. So that's why when somebody comes in and wants to do a project with us, we always have to do 12 tests. and usually every color range is specific to each project because of the material. We also have a physical database in the lab. And then it's also about, thinking out of the box and because it's kind of an unknown world in design and art, scientifically not, but then it's looking into shapes. For example, if we have a 3D Petri dish, how would that affect the color grown into it? The last research we're doing is creating some paint based on different sizes of silicon nanoballs that are then covered with melanin. And then you get this iridescent paint. It's a lot of work for a couple of milliliters of paint, but yeah, it has been fun so far. And then you can also apply it into paper. We recently also published an online website on which we also have a database of all the research we already did with several materials. And there we also showcase storytelling projects. So I'm the designer of the team and responsible for storytelling because, for example, structured colors, not many people know about it. And so I kind of tried to talk about the story of what are structured colors, where are they in from nature? And then I tried to also apply the techniques in the project. So this is Entomocolor, which is a collection of 12 beetles that are critically endangered and extinct in Flanders. And they are threatened because of there are biotopes that are threatened because of global warming and extensive farming. And we kind of try to recreate those colors to talk about a lost color palette. Voila, this is some fabrication. And then we also work together with artists. There's a collaboration with Anne-Veronica Janssen. For us, it's always interesting to collaborate with artists because they usually have a specific expertise on a material or technique, and then we apply our color research onto it. So this is, she really pushed us to get out of the small size of the Petri dish, and these are samples of 60 by 60 centimeters. So this was almost the biggest we've grown. Now she also made some pieces of 1 meter by 1 meter. And here you can see that the color is not just one color, but you really start to see different shades of color. And this is because the surface tension in the boxes where we grow it is different because of the size. Voila, that's what I wanted to show you. Thank you.
Thank you, Heleen. I've formulated some questions beforehand, but I've adjusted some of them and found others. So I'll try to find what I was thinking of. To start with, what intrigued me very much is effectively, so Adrien is talking about parallel worlds, and it's really great that today we're looking much more to animals, learn much more, and also think much more about cohabitation, as especially Adrien and Sebastien have showed in their research and how they work with the results of that research. But what struck me was how small actually the range of human visibility or colors that are the very extremely small range of colors that humans can see normally. Sebastien showed this table where there's really this really tiny little bit that we humans can see, and I wondered if any of you have actually ever thought about why there is such a huge range of colors, because it's wonderful and you've shown it, but any ideas why it's so fast? And I also wondered whether there's a specific reason or difference between fluorescent and iridescent colors. So because there's one thing that there's color, and how is it produced, and so why is there such a wide range? Might be a question that you can't answer, that's possible, but it intrigues me enormously. Any of you?
Okay. Yes, these are complicated questions, not necessarily easy to answer in a few seconds. But for the wide range, first of all, as Adrien mentioned, the visible range is from 400, 380 to, let's say, 700 nanometers. But okay, colors is much more than only the wavelength, but it's part of it. But that means that for every single wavelength, there would be potentially a different colors, different color that could be produced. We can also combine them. So there is that's a continuum. So there is an infinity of wavelengths between these two values, if you understand what I mean. That's yeah the first answer. Then I think that there is all the perception of it, the brain interpretation that comes there, where, well, it's probably outside my my expertise. Yeah, what was the evolutive advantage that we found to perceive all these colors? That's a fairly good question.
Yeah. So, well, you mentioned, but yeah, no, go ahead, Adrien.
No. Yeah. No, indeed, it's wrong to think that light is equals color, because the light really comes to us, then we are sensitive within the eye to certain segments of light. But then after that, there's like a very complex process of interpretation that is happening. And for example, if this process was not happening, for example, we would be completely unable to distinguish between like transparent objects and opaque objects.
Yeah.
Because a transparent object, you can, if you take a photo of it, becomes all flat, and you can interpret it as a flat painting that looks like an aperture also, you know? So, okay, we have a limited range of colors, yes. These are actually well defined. One of the diagrams that Sebastien was showing shows the limits of colors of what human beings can see based on these compositions of light. But then we have many it's a bit like, okay, color would be like one level, and then there's like chromatic levels, if you will. You perceive, with the eyes, you perceive also indirectly your sense of touch. And, when you see an object, you can tell if it's smooth or if it's like grainy, and all these things can get them through the visual sensations. So,
yeah, go ahead.
Yeah, and then about there are some famous things, like most apes are dichromatic, so relatively to us, they are colorblind. And when humans started to see red against green, they probably became very good at picking some fruits. And so, of course, there are like some kind of random evolutionary situations where the individuals that are much better at picking up the fruits will be much better at surviving, etc. But
is there still a question of evolution, because you both mentioned this in your answers? So is it a static given, or do you think there's still some kind of evolution going on in the way that we humans can see things? Because it also brings up the notion of visibility. When Sebastien talks about bird-safe glass, it is because birds can't see the glass. But in the other way around, we don't see a lot of things that animals can see. So which would almost question whether there's actual things that birds can, or any animal can see, that we can just not perceive. So do you think that there's still an evolution going on, or are we completely dependent on artificial?
I mean, I hope so, because otherwise we are like a dead species, you know, like.
Yeah.
But yeah, like they are like, for example, in many flowers, there are like UV patterns that we can't see. So like a yellow flower will have two colors for B. There are many situations where basically, in my opinion, what we call color blindness could almost become like our definition of color, because we are limited by definition. And so relatively to another individual, there are already slight differences. And relatively to another species, there's a lot of differences. And not only in terms of color, but like movement detection, resolution of the sharpness of the visual system, time resolution. Some insects are like maybe 10 frames per second, more or less. Bees have 200. So it's all the parameters that we can measure of the visual system. We can imagine that if we have the value for human, this is not universal, it's a particular case.
Yeah, it's maybe, I mean, I'm not a specialist at all in this, but so I think that Heleen, on the website of Ecology of Color, there's mention of the well, you mentioned it also in well, more or less all of you talked about structural coloration, and you give on the website the example of the peacock, which feathers are pigmented brown or melanin, but that it's only through the light that it reflects as blue, turquoise, or green, depending on the angle. So that was quite clear in all your presentations how that works. But how does the peacock perceive that themselves? I mean, it's difficult to, because it's fine that we humans can sort of find out how it works, but how do peacocks or any of the animals that were mentioned perceive them actually? Any idea?
Yeah, was Sebastien mentioned that most of the birds have an extra cone, and because of that they can also see ultraviolet. So I've seen like an image how a peacock sees the other peacock, and it's like a really like a dark purple, and then the eyes and the feathers are like really sharp, and they're this bright purple.
Yeah, I suppose that is based on very material ways of looking how the lens works.
Oh.
We've lost Edith.
We lost Edith.
You disappeared.
Yeah, I noticed that you were all standing still, and I was probably standing still for you. So I don't know. What did you get for my question? Nothing.
Yeah, say it again.
Well, yeah. So again, a very weird question, maybe for you, but I just wondered how it was discovered what animals can see. How is that?
I think by observing the retina of some animals, you figure out that they have different photoreceptors, cones, and you can measure the absorptance of these cones, and from this we can infer to what they are connected. There are also further studies to develop these vision models that I mentioned too. And if I may use an engineering metaphor, that's how they are connected in a way. There are opposition processes to allow the vision in the living organism and figure out how the signal is processed. Maybe to also react on the previous question. That's complicated to see to figure out what birds would see with that extra photoreceptor. But one example I saw at some point on internet was tigers. When you have a tiger in a green environment, for human being will spot the tiger easily. That will be something that is black and beige, let's say, in a green environment. But for our animals that are potential prey and are bichromats or slightly different, actually they will be very well blended in the environment. And well, having that extra photoreceptor, being able to have a green and a red photoreceptors allow us to spot easily a tiger. Just imagine a tiger that is green and black, and that's what other animals would see.
Yeah, and it's fine
they won't
for
see
them.
it.
Yeah,
Exactly.
Yeah. Well, in that
So
sense
sorry,
also,
no, sorry. I just want to add, because I don't agree with one thing you said. To know how do we know what the animals see? Way before studying like the chemistry and the optical pigments in the eyes, there are some behavioral experiments.
Of course.
Really how we know most of the things, and even the thing that we think we know, they have to be validated by experiments.
Yeah,
so
totally.
for example, we know that the bee can see some colors because we use color patches, and we train the bees to find sugar on a certain color. And if they find it immediately, it means they see that color. If they hesitate between different patches, then it means they probably confuse this color with some other colors.
Totally, that's another way to do it. Yeah, you're right.
Yeah.
That's another way to do it. Yeah.
This started at the beginning of the 20th century, this kind of experiments. And there was even one guy who started to try it in the 19th century, but he used like red objects, and bees don't see red. So the experiment was a failure. But so it's always by training and playing with the animals that it can be really known that and animals see colors, so they can associate like us the colors to an object. If you change the light, the color changes for a camera, but the animal is still able to recognize the object by the color. The animals have color most animals have color memory as well, and it's really it's not a joke, it's really colorful.
No.
This being said, dissections of animals and visual systems started fairly early, and optical microscopes are known for a long time. So I think that both approaches started fairly early. But indeed, behavioral, also with fish, works fairly well.
Yeah,
you're right.
Yeah, in that sense, I've been a long time intrigued, but from another point of view, more as a curator, how and the connection with the animal world, how Australian bowerbirds, when they build their nests, they select color elements to attract a mate. And normally these are natural materials, so berries or leaves. But when they are close to human habitats, they might use also waste. And there's a funny thing where they might either have really a predilection for one color, either blue or red, which is quite extreme, I would think. But clearly animals also have a preference, which is maybe difficult to indicate why it is exactly. In terms of a very, well, always kind of anthropocentric approach to animals, I wondered, Sebastien, you were talking about the bird safe windows and the patterns that you make on them. But the patterns, are they also, where do they come from? How do you decide what kind of patterns?
So far, we simply made some patterns that looks like an obstacle for a bird. So one is simply a grid pattern, plate pattern, and the other one is a bit more random. But yeah, indeed, from that point of view, we can surely improve. The idea was simply, okay, a bird should identify this as an obstacle so that it will slow down and avoid a collision, or a strong collision with the object that it will perceive.
Yeah. I think Adrien, and then I go to Heleen. You are currently, so you're having 2 residencies, I guess. So the place where you are now is for work that you briefly showed something that you were doing there, and then you are also in Gluon in Brussels. And what are you doing there?
Where?
Is that in Gluon?
In Gluon, I'm working on what I presented about the insect vision and
Okay, yeah.
So I'm trying to come up at some point with a proposal for an installation where we would have some kind of experience that resembles this kind of perturbation that the moth experience with our lights.
Yeah. And since I'm based in Rouen, where there's a festival on Impressionism, and you told me that you will be present here as well, and that will be more with the glass, or
Yes, I'm gonna make a glass piece. Well, I can show you quickly, like so I'm making these glass pieces with these machines that we have like made. You see the machine is working right now, and so they are like, you see this map. So there are 96 panels of that size that should be done within the next 5 days. And in Rouen, I'm gonna make a window with the same technique, but probably with some color. So I'm in a glass factory right now. Like these are kilns, like large scale kilns. With it's 600 degrees in there. It's currently being the glass are being heated up, and this is like a part of the no.
Okay.
You see now? Yeah.
Yeah, we can see it. Yeah.
This is like the piece that I'm working on. It's quite monumental.
Yeah.
Yes.
And how is color involved in that?
Well, in this case, it's only different sort of thickness of the same white material.
Okay. Yeah.
It creates like sometimes the white is seen as a shadow, sometimes it's seen as like a diffuser, so it becomes really bright. And when it's more diluted, then it can become invisible or reappear when the sunlight is shining on the window. So these are like, it's also like a form of playing with the appearance of the material, but not really with the spectrum of light, but more with the intensity, the direction, the shape of the source. Because, for example, when you're in the shadow, it's the sky which illuminates the piece. So the light is coming from every direction around the piece. But when the sun hits the piece, then it's directional and the appearance changes dramatically. And so I'm playing with these kind of parameters of natural light.
Okay, yeah. Heleen, you were talking about, well, or you mentioned anyway, the instability of the colors that you found, the living colors. So you make a positive of a negative, so to speak, by saying, well, it fades, and that is part of what it is. And then you also mentioned that it's a lot of work to get a bit of paint. So this kind of research is you well, you work in design, so in the lab it can be small quantities, but if you want to apply that to a larger industry, how do you go about that? Is there a connection that you make there, or how do you research that?
Yeah, now I also realized I forgot to mention something really important about the structured colors. So basically, when we grow them based on melanin, we have, I mean, the color is staying forever. So it's very stable. But on the other hand, and that's the same what happens if you touch the wing of a butterfly,
Yeah.
your finger turns black and you erase the 3D structure.
Okay.
So that's, for example, like, yeah, it's very fragile in that way. And
Yeah,
for that, it's so lucky us that we are in an art school and that sometimes, yeah, artists approach these things in a different way. Like, for example, Lucca, who was about the fragility of and so on. I think so. We are part of the design department, and the nice thing is that the other courses, they are like small little industries, of course, way more experimental. We were approached by a printing company to develop a kind of inkjet ink with fading colors, for example, for food packaging and so on.
Yeah.
And then, yeah, we started trajectory, but quite soon we discovered that you actually need to have a whole team of scientists and PhD students on it, and we don't have the resources at the moment.
Yeah.
So that's why we always do kind of proof of concept, and for example, the band plotting, but to actually get it into a printer, especially the inkjet, it's not that easy.
Mm-hmm.
And then with the structure colors, yeah, we mainly have the issue of touch, but I kind of identified that it would be really nice to, for example, work together in some more niche markets, which are more speculative. And then I'm thinking about fashion design, for example, but also about car design, like how would the future of a concept car look like? How would the car industry be in 100 years? Would we be coating in bio-based materials and so on? But it's very, very hard to reach those people. I mean, we've been doing some efforts, but so far, no luck yet, but we keep trying. Yeah.
And in terms of the paint that you develop from algae, because that is also the question of quantity.
Yeah, so with algae, it is, as with biotechnology, you're able to control the parameters of growing it. And there are also, especially in Spain, for example, and the more warmer countries, there are a lot of algae farms. And algae is also used for biomass, is also used to purify water. So yeah, that is possible to upscale. But then, of course, yeah, you need to find people that have the same values as how we approach the fading. How can you see it as a smart ink and not as a mistake? And that's usually how industry sees those things more traditionally. Like they do everything to keep the color, like forever. Well, we see, for example, that working together with students, we see that nowadays, for example, with young art students, it's not that important to have their signature forever, but they're also open to art that disappears or that is completely ecological. So that's a nice shift to see.
Yeah. Do you make the link to the industry at all? I mean, you work large scale, Adrien, and I don't know if Sebastien has any connections to translating your knowledge to other ways of applying it.
Yes. We have some contact with the industry. We work also with some industries. Well, for the bird glass windows, there have been some interest from some companies, but also on other applications. For instance, I didn't mention color change, but some beetles, butterflies would change color upon contact with liquids. For instance, the blue morpho, if we drop some water, ethanol on the wings, they will change from blue to green. I see Heleen nodding with her head, so it's a fairly known effect. And so, well, the inspiration is obviously some gas sensors, vapor sensors that could be made, or smart windows that change, that are responsive to the environment. And these are also type of applications that we've developed in the lab.
Yeah.
Yeah,
And
Heleen, I was wondering if did you try adding some? I mean, there are some additives that maybe could be also biosourced. I don't know, but to like block the UV and because like the yellow fading you have is like quite a common thing with dyes.
Yeah, with the
And
painting.
of course, like many additives are like quite nasty, but but maybe they're also like more natural or more like safer types of additives. And because it's interesting, I really understand your point of having this kind of time programmed colors, but
Mhm.
it could also be interesting to have something that's sustained at least a few months or.
Yeah, but for example, the blue and the purple, they're way stronger. Eh?
Yeah,
So
but
it gives you the clarity.
they reflect the photons that are the most damaging. So the
Mhm.
yellow absorbs the blue and the violet, and
Yeah,
these things
yeah,
are more energetic. So quite often it breaks faster
yeah,
than yellow things.
mhm.
yeah.
Yeah.
Well,
And so they are like they are the products that scatter or reflect this part of the light without
Mhm.
changing the color. And these are like common additives into like inks and things like that.
Yeah, no, we haven't done research into
Mm.
that.
Adrien, are you in contact with, or maybe not yet, but with the light industry, for instance? I mean, you talked about this article in which you found some mistakes. I mean, this is quite important in terms of how we see the world today and the disappearance of insects and birds. And
Mm.
since well, we hardly have any undisrupted night light anymore, especially not in Europe. Do you envisage working with them or
Well, I'm in
not really?
contact with a lot of different companies. So for example, for this project, one of the outcomes I want also to make an LED which is not human centered. But for this, I usually don't like to work with big European corporations because they don't answer the phone, or you find a marketing department and it's always
Yeah,
very annoying, or
yeah,
they have no idea what we are talking about.
yeah.
So I only work with companies where I can talk directly to the people in
I
the
can
lab,
imagine. Yeah,
engineers. It's much better.
yeah.
And for the LEDs, anyway, everything's in China. So
Oh yeah, as
then
well.
I work with the Chinese people, and they
Mm.
are very good.
Okay. Yeah.
And
Well,
yeah, but yeah, I mean, in my position, this is what I do. I'm like last week, I made this custom-made inkjet ink for my glass project here. It was made in Spain on request, and I always work like this. When I need something that I can't produce myself, then I go directly to the manufacturer and we design together a product.
yeah. So in that sense, you can also develop, but maybe on a small scale, and the people that you get on the phone
Yes,
directly, which I understand completely, a light or LED light that is
yes,
uh,
exactly. Yeah, because if you call you know who's manufacturing LEDs like in Europe, well, no one. But let's say you call Signify or Philips or
yeah.
they will probably say, yeah, if you want this, you can order 300,000 units.
Mm. Yeah.
And then you need like 500, like, yeah, maybe 300,000 euros then, you know.
Yeah. Exactly. Yeah. Well, in that sense, I mean, it's an interesting problem that probably from all the and I think your research is really fantastic of all three of you. It was really an eye opener for me, and I hope for many others. I don't know, Alexandra, do you have any other questions? Because of all the questions I prepared, you actually mostly answered them in your presentations. So Alexandra, do you have any questions?
Well, I don't have any questions, but I really liked presentation of everybody because I think it gives some kinds of the very interesting, more or less, impact on for especially our artistic society, just to know how color we can work and what it can bring and what kinds of another topic it will more or less trigger, especially when I'm talking about that our vision is not really vision like all another beings on this earth. So, but asking question when there is a presentation so rich, for me it's very difficult because there is answer already in it. So
yeah, for me it's as said also indeed the way of how we can put ourselves in the place of non-humans, which and find our proper place on the earth. That is the idea of parallel worlds or cohabitation. I don't know if there's any questions from an audience that is looking, but otherwise Ekaterina would put it in the chat, and there's nothing. So probably everyone is stunned by all the information that was delivered by you. Do you have any finishing remarks, Heleen? You not?
Yeah, I have a question.
Yeah, go ahead.
Because currently I'm doing some research in the lab. So basically, I'm a maker. So I always start to make, and then it's like, what do I do with this? So lately I was experimenting with titanium and anodizing, and I got beautiful colors, really bright colors. And then it made me think of tropical fishes. And then I read this article about tropical fishes in the ocean that because of climate change and because of the heating of the water, they have to swim a couple of meters lower. And that means that they see colors differently and that there are troubles with the species recognizing each other, camouflage and so on. I was wondering if you have any expertise on that, or I mean, I really just started the research yesterday, but I was wondering maybe you have a point of view on this, or somebody I should look at.
There is in the article that I studied the most, there is one of the authors is a Belgian guy who works in the UK. His name is I don't know, sorry, but he's in the same team. Let me find the name of that person. And his specialty is like the color vision, but mostly of like submarine species. So that person would probably answer all of your questions.
Great.
If I may just comment on this, last year we published with colleagues from Australia and etc.To a study where it was not about the visual system but more about the color appearance and due to melanin. And actually we checked the effect of reactive oxygen species on the melanin of some fish species in the Great Barrier Reef. And well, that affect the optical properties of the melanin, and so basically due to oxidation of melanin by reactive oxygen species, potentially the fish appears differently too, which may raise some problems for identifying other mates from the same species to avoid predation or sort of things. Actually, Swiss. Cedric is Swiss. Adrien.
He's Swiss. Oh yeah. Well, sorry, he has such a Belgian name.
Yeah, his name is very Belgian.
Well, in terms of that question, because you talk about climate change and we've earlier talked about evolution. So I wondered whether there's maybe also something that might change. So you talk about fish that have to swim deeper, but maybe there's also a way of adjusting colors in answer to climate change. But I don't know if there's any research into that yet, or...
I mean, the biologists are studying adaptation of, for example, like I forgot his name, but one of the person I'm discussing with right now at VB, he's setting up an experiment now about like the impact of artificial light on some insects.
Yeah.
And the study is really to see if they can adapt to it or to different conditions, to different types of light. And yes, so like the same way as we adapt all the time to our own things that we do, they have to adapt to this as well, because we are dominating the planet quite a lot.
Yeah.
Well, until it starts dominating us back, but yeah. And so they do adapt, yeah, of course, all the time. Or they die. They adapt or they die. So
yeah, exactly. Yeah. Well,
I had one more question for Heleen. I was wondering so not the structural color part, but the dyes or pigments. Do you have like, I saw you have like quite a nice material database. Do you also have like a spectral database? Did you measure this with a spectrometer or something?
No, I don't think so. With the research before me, no.
Okay. Because I'd be very interested to check some of the colors in this way. So maybe if I have time once, I should come and
yeah, you're always welcome. And I'm also really curious on how, I mean, because the structured colors are really nice because they're experiential, because of the iridescence, and you invite the spectator to look, but then if you start adding different light sources like you do in your work, I think it's can be mind-blowing. So
but I'm really talking more for it's really about more like the stable, the classical looking colors. Because the reason why I started to check like flower petals, for example, is because they have sometimes absorption in areas that interest me and that I can't find in the synthetic products. And so I'm thinking, like, do you have, for example, like a very whitish turquoise looking pigment somewhere? Something that's like turquoise blue, but a bit whitish. You have this in mind?
I will note the question then for my colleague Maria. She's a scientist and she knows all about it. I mean, it's more the previous research.
Okay,
yeah.
I just wanted to add something in my previous explanation about reactive oxygen species. What I forgot to say is that it increases due to climate change and anthropogenic pollutions. That's what makes sense in the conversation. Sorry about that.
Oh yeah. Well, I'm glad that there's some connections made here, and maybe we should all visit each other. But I really look forward following up on your research and work. So please do keep me posted. And I think we can finish our conversation with this. Thank you very much for your very rich contributions.
Thanks to you.
We'll look forward to meeting you in person next time.
Of course.
Thank you.
Yes.
Thank you.
Okay. Thank you.
Alexandra, we can't hear you. Alexandra, we can't hear you.
Okay, thank you very much. It was really a big pleasure to have you here. And also about evolution, there is some book, but it's already 2007 that it was edited, but it's just in general about vision. And there is also a very interesting article, very contradictional about color. What is neutral color and what is for human being, and how we're more or less distinguishing it. But okay, so just it will be otherwise very long. Thank you very much for everybody, and I hope that we will see each other soon. Thank you.
Okay.
Thank you.
Bye-bye.
Bye.
Bye.
Thank you, our team. Thank you,
yeah,
our
as well.
technical team, just to work and just to stream everything on the YouTube because it was really good that everybody can follow it. Thank you.
Okay, bye-bye.
Bye.
Bye.
Bye.